if any of you know the story, it was the 1970s. It sort of had a cult following as far as radio programs. It's a comedic sci-fi series that talked about the misadventures of the last surviving man, Arthur Dent, following the demolition of planet Earth to make way for hyperspace bypass. So the Earth was accidentally destroyed. This guy was the last guy on Earth, and he met this sort of humanized robot that allowed him to hitchhike along the way so he could discover the secrets of the galaxy. And so in many ways, I feel like the cell looks a lot like the galaxy, and many of the things that we study in terms of gene networks and protein networks actually resemble aspects of the galaxy as well. So I want to tell you today about kind of my journey as a hitchhiker in science and how I have really come to learn a great deal through this process, through integrating myself with other scientists and really forging ahead in many new fields. Here's my hitchhiker's guide to programming and reprogramming cell fate. So I'm going to talk to you, give you vignettes that are somewhat based from the work in my lab, although I'm going to make it somewhat data light because I really want you to understand the concepts and where we've taken those concepts to sort of our final lesson. So I'll talk about DNA dark matter, which is this DNA that was once considered junk DNA, and how we now know it comprises a vast universe of regulatory sequences controlling cell fate and disease. Lesson 2 along the way has been that also this DNA dark matter encodes thousands and thousands of non-protein RNAs, non-protein coding RNAs, that also play roles in various biological processes, including cell fate. And I want to point out that these lessons have really only come to light in the last decade on a large scale. And also that they are fundamentally the outcome of improvements in high-throughput technologies. And lesson 3, I will tell you about how we've exploited what we've learned from our analysis of what drives cell fate to understand how it can be reversible in certain instances, such as the case of cardiac regeneration, as Nadia just mentioned. Okay, so my lab has been primarily interested for a very long time in how the genome of a single cell can give rise to the derivatives of all 3 germ layers. And so this is a very complex question, a complex problem, and this problem has been studied in many animal models, including mouse, and zebrafish, and others. We chose to study how cells make decisions early on in embryonic stem cells. There are a number of reasons why we chose embryonic stem cells, and now we also use induced pluripotent stem cells, which I'll talk about in a little while, because they can be derived from this inner cell mass. They can basically be a freeze frame of these early developmental processes, and in response to developmental cues, can then be turned into these different cell types. So very carefully we can track some of the decisions that they make and some of the key circuits that drive these decisions. And then of course, lessons that we learn can be ultimately tested in vivo. So at this point, when we decided to embark on an understanding of what drives early cells to become the cardiovascular lineage, it was very clear to us that our DNA was so much more than we thought. In fact, only 2% of the genome codes for protein coding genes. Yet we have this capacity to generate hundreds of different cell types with amazing complexity at the right time and right place during development. How is that possible? Well, lesson 1. I'll tell you about some of our studies to define on a broad scale regulatory sequences throughout the genome. And this is also basically comes from the Galaxy's Guide, as I told you at the beginning, by Doug Adams, where he says space is big, really big. Well, so is cell complexity. And so how do we approach this? Well, for many, many years, we've known the role of transcription factors as key for regulating the gene networks that orchestrate cell fate. And in this example, and in the examples that I'll give for the rest of the talk, I'm going to focus on our work on cardiac biology. As you know, the heart undergoes amazing morphogenic movements as well as restructuring to make this beautiful functional three-dimensional organ. And this organ actually has to pump blood throughout life. Okay, so this has to be probably one of the most magnificent machines that were ever invented, for sure. But this is not an easy process, and it's driven by sets of genes and transcription factors and networks that fundamentally drive this process. And we know that even subtle perturbations in these networks can lead to a whole host of congenital heart defects. And this is really important to understand. How do these factors work? How do they coordinate with each other? And how, when mutated, do they lead to congenital heart defects? Because it really is the primary cause of infant mortality and morbidity. And to put it into perspective, every 15 minutes a baby is born with a congenital heart defect. So we're very interested in trying to understand how these factors drive these various decision-making points. And so I told you that most of the genome does not code for protein-coding genes. And so really, we don't understand how they're regulated on a global level in response to signals. There were studies over the last 30 years that suggested that these distal DNA sequences, called enhancers or transcriptional enhancers, could play key roles in regulating genes. Although before the advent of high-throughput technologies, enhancers were very difficult to find genome-wide because they lack a certain conservation. There's no way that you can just see the sequence and say, aha, that's an enhancer. And they also reside at variable distances from the genes they regulate. So it's a bit like finding a needle in a haystack. However, enhancer regulation of genes is really key to developmental complexity. So we and others were able to identify some patterns in chromatin that were very critical for starting to be able to identify potential enhancer candidates in a tissue-specific manner. So Joe in my lab had figured out that histone modification patterns could actually be a proxy for looking at enhancers that were active in a particular tissue. And if you then look at these sequences, you can actually predict transcription factors that bind these sequences. And then, as I'll talk about later, there are other ways also to predict their target genes. So not only does the ability to perform something like chromatin immunoprecipitation genome-wide and to identify these sequences followed by computational analysis of potential transcription factor binding sites, it gives you these nodal points that perhaps drive particular cell fate decisions, such as in the heart. But it also now gives you sequences that you can go back and look and see whether or not mutations or changes can lead to an effect on these gene networks, which I'll talk about in a minute as well. Some surprises is that there are tens of thousands of hundreds of thousands of enhancers that are active very specifically in each particular cell type. And so being able to identify these particular active enhancers tells us a lot about any particular cell. In fact, you can predict the cell type just from understanding the active enhancers in a particular cell type. So in order to basically understand how enhancers might be driving or orchestrating gene programs, we developed a method in order to take pluripotent stem cells and actually drive them to be terminally committed cardiomyocytes, but during this process being able to identify and isolate key points along this trajectory that are akin to in vivo time points during development. And so at the end of this protocol, we can get a nice dish of beating cardiomyocytes that we can then identify potential sequences that can drive this process as well as the function of cardiomyocytes. So we analyzed these patterns, we identified enhancers, and then from this, we took these sequences, and I just want to show you an example of the kinds of meta-analysis that can come from this. For example, you have these sequences, for example, this is in cardiac progenitors or precursors, and these sequences are enriched in a particular transcription factor binding site called MEIS1. And if you actually ask what these enhancers might control, you find a whole network of genes that regulate cardiovascular system development. So now just in one fell swoop, you now have a candidate transcription factor that you understand what it drives in terms of processes. And what's interesting about this as well is MEIS1 had been knocked out in the mouse prior to this and showed important phenotypes, although those phenotypes were not understood at the molecular level. This is also the case here for another example for GATA, where we can find genes that are associated with these enhancers that drive particular gene cohorts. So the other really interesting aspect of this is we're able to then look at how transcription factors can combinatorially regulate different processes compared to when they're alone. So I don't want to go too much into detail than this, other than it really opened up the entire field in order to be able to try to understand how specific decisions are being catalyzed by these enhancers. The other aspect of our work really led us to start to look at, or go back and look at these genome-wide association studies. So for many years, the NIH has been supporting genome-wide association studies, millions and millions and millions of dollars, really to very little avail, because it turns out that most common variants across the population don't fall into protein coding genes. So how do we predict how some of these common variants affect disease processes? So we and others had the idea that, okay, if these variants are located in loci that do not overlap protein coding genes, could they overlap these enhancers? You can imagine that this here is called a Manhattan plot. These are the significant signals. This is, for example, a chromosome, and this helps you to zoom in on a region of a chromosome that may be important or associated with a disease. In this example I'm showing you, for example, you could just get a sense that there's no genes here, so how do these variants or this signal possibly tell us something about what's happening or contributing to particular diseases? So we took as an example a cardiac trait, and we asked all of the loci that are significant, that do not overlap genes, are they enriched for these active cardiac enhancers? And the answer to that is yes. But how do you study this? So let me just go through this quickly. Xinchen Wang in my lab actually developed this methodology where he could take and impute these SNPs, or common variants, across the genome. He asked which ones were actually located within cardiac enhancers. He mapped them to candidate genes that they perhaps interact with, and then he was able to show which variants actually affected which genes. And then within both mouse data as well as work that we did in zebrafish with folks at MGH, we could identify a downstream gene that was affected by a single nucleotide change in an enhancer that disrupted a transcription factor binding site. So this really opened up the whole world of epigenomics. And now we can use epigenomic signals to now better understand how cells make decisions and how faulty regulation leads to disease. So, lesson 2. So this is something that was from this book as well, where I think it's kind of funny because everybody is a genius, but if you judge a fish by its ability to climb a tree, it will live its whole life believing that it is stupid. And that's what kind of leads me to this next point about non-protein coding RNAs. So only again because of this advent of high-throughput sequencing and our ability to interrogate the genome in ways that we had never imagined possible, did we discover that there are many, many transcripts that are being produced from regions of the genome that do not code for genes. However, these long non-coding RNAs, as I will refer to them for the next couple of minutes, actually look a lot like mRNAs, but we can't treat them like mRNAs. They are actually 5' capped like an mRNA. Oftentimes they're spliced, and they can often be polyadenylated. Usually we just have a cutoff of 200 nucleotides to call them a long non-coding RNA to separate them from the smaller class. But quite, strangely, they do not have significant open reading frames. So what are the function of all of these RNA species throughout the genome? Are they just random noise, or do they actually have functional roles in processes such as development? Well, we became very interested early on in this problem because there was evidence that they were potent specific regulators of gene expression. They displayed cell type expression patterns, but what really kind of perplexed us was that they're poorly conserved at the sequence level. And I'll get back to that in a minute. So these three folks were instrumental in identifying the first long non-coding RNA with a role in cardiovascular commitment. And to kind of go through a lot of data, I will just illustrate in this cartoon, using our ES cell differentiation system, we were able to show that Braveheart, which is a non-coding RNA of about 600 nucleotides and 3 exons, is critical for the progression of undifferentiated mesoderm to cardiac mesoderm. And when you lose Braveheart, you don't turn on the who's who of transcription factors that are required to progress toward terminal differentiation. But at that time, we really didn't understand how it was functioning. And as I mentioned, these long non-coding RNAs tend to be poorly conserved. That doesn't mean they're not important. Let's think about RNA for a minute. RNA is very functionally diverse. You can actually have many different sequences that form similar structures. And so from that perspective, we wanted to understand whether or not there was something in the structure of Braveheart that could tell us something more broadly about its role in cardiovascular commitment that might be extrapolated to human. So we performed some chemical profiling, and through a number of experiments and careful analysis, we identified the most likely structure for Braveheart. You can see it's modular, like many RNAs. We noticed that there were a number of structures. Many of these are represented in other RNA species. But we noticed there's this 5' structure, which we call the asymmetric G-rich internal loop, that was not well represented in some of the RNAs that, such as ribozymes, etc., that had published structures. So in addition to mutating some of these other structures, I'm going to focus just on a little bit on this agile motif, because mutation of this little loop here actually gave us quite a striking phenotype. So using CRISPR-Cas9, we took out the 11 nucleotides that made that loop. Taking out those 11 nucleotides diminished the loop but did not affect the structure of the remainder of the RNA. We performed our analysis in terms of trying to understand how now, if you had normal expression of Braveheart, except you did not have this loop, what would be the phenotype? And much to our surprise, similar to complete loss of function, we saw that these cells right here failed to really downregulate brachyury, which is a marker of undifferentiated mesoderm, and could not turn on the who's who of transcription factors, as I showed you before. So to us, this suggested that this small loop had a functional role in regulating cardiovascular commitment. So agile became quite interesting to us, but again, as I told you, this entire transcript really didn't have a human homologue. So we took the approach of asking, what does this loop interact with? We found that it interacted with a transcription factor called CNBP or ZNF9. Interestingly enough, CNBP is the only transcription factor that's been implicated in myotonic dystrophy type 2. And in these patients, they often have heart defects cardiomyopathies, etc. So we've gone on now to ask whether or not CNBP interacts with other RNAs, not only in mouse but also human, that look like Braveheart. And I'll tell you, we have a number of candidates suggesting that this small region actually may be the functional aspect that can be extrapolated to similar functions in both mouse and human. And you can imagine that the importance of identifying an RNA structure may have very important therapeutic potential. So here's lesson 3, and I'm going to go through this quickly because I'm running out of time, which I think is been the surprise of the last decade, that cell fate is reversible, and how this has strong implications for tissue engineering and regeneration. And so again, I like to try to bring something from the book that I started with. It's a mistake to think you can solve any major problems with just potatoes. But what does that mean? So that means that there is the complex nature of trying to understand cell fate, that we need to take a multidisciplinary approach to understanding the problem of cell fate, regeneration, and how we can use that for therapeutic purposes. So, as you just heard, the heart has a built-in obsolescence. It's remarkable. It's made to work and pump throughout life. However, as we age or in response to disease, we can lose billions of cardiomyocytes. In fact, after a myocardial infarct, you can lose up to 1/4 to 1/3 of your cardiomyocytes. And the turnover in the heart is insufficient to replace the injured myocardium. And as you just heard, and there was a discussion about it after Nadia's talk, there doesn't appear to be a resident stem cell population that's giving rise to the new myocardium. However, current evidence suggests that cardiomyocytes can de-differentiate or reverse their fate, re-enter the cell cycle, and make more of themselves. Obviously, they don't work in isolation, and I'm in full agreement with the last speaker suggesting that signals from the immune system as well as fibroblasts are absolutely key to signaling these cardiomyocytes ultimately if we want to promote regeneration. So a couple of things that I just want to point out. Our heart continues to develop even after we're born. So during embryonic development, our heart grows by proliferation, and we use glucose as a primary carbon source, and our cells are mononucleated. But this remarkable thing happens, and I'm just going to use the timeline for mouse development as an example. Postnatally, during the first week of life in mouse, cells go from proliferating to growing by hypertrophy. All of the cells exit the cell cycle. They become binucleated or polyploid, and you have this dramatic shift in metabolism, and now you've limited regenerative potential. So this provides us a great opportunity to try to understand the molecular roadblocks of regeneration. And once we understand these molecular roadblocks, can we leverage these findings to actually manipulate cells in vivo ultimately to promote proliferation? So one of the things that we showed a couple of years ago is that this idea of de-differentiation is actually observed at the transcriptional level. Genes that go up or down during maturation actually go the opposite way in response to injury. So this was a really good point for us to try to understand some of the factors that were controlling this process. And so because we've had a lot of experience looking at chromatin structure, which really, whether it's compacted or open, defines where transcription factors bind, we chose to look at a series of chromatin remodeling enzymes to try to understand whether or not they were involved with this process. To make a long story short, we found a particular gene called RUVBL2 that showed as much increase in proliferation as some key positive controls that had already been shown in the literature to promote cardiomyocyte proliferation. Why is this important? Well, this is the first time that RUVBL1/2 have been implicated in maturation of a particular tissue. There are now links to perhaps liver regeneration, but these genes are code for triple A plus ATPases, and they are catalytic components of chromatin regulatory complexes. About 12 or 14 years ago, the genes that are homologs in zebrafish had been implicated in heart growth, but really not much more had been done with these genes. So we've created some loss of function neonatal cardiomyocytes, and we've looked at the transcriptional changes, and I just want to show you what's really fascinating about this. As I told you that during maturation, you have dramatic changes in metabolism, and you also see increases in sarcomere components as well as other cardiac genes. Well, here, when you lose RUBBL2, you see the exact opposite. We're particularly excited about this because it really suggests that it's a master control switch for regulating maturation. And we are now looking at this in mice in response to injury, and I hope to have more to tell you about this soon. So just for the last minute, I'm going to tell you about sort of the final lesson here. I told you some vignettes about how we discover what regulates these important gene networks during cardiac development. But really our ultimate goal is to be able to leverage these developmental principles, form equals function, for tissue engineering and regenerative medicine. And if you asked me 10 years ago if I would be doing this, I would not have known that that's where I would be headed. And so I may not have gone where I intended to go, but I think I've ended up where I need to be. And one of the messages that always makes me laugh in the book that I had told you about, that whenever you're faced with a new problem, their piece of advice is don't panic. So that's what I'm going to do. I'm going to get excited about the potential of all the things we've found. And it puts us in a tremendous position to engineer cells. All the new tools, some of them you're going to hear about this afternoon in Ed Boyden's talk, also in Jim Collins' talk, how we can manipulate these networks, these cells, to create allogenic cell types that can perhaps be transplanted, but more importantly for the things that I'm interested in, how mutations during development leads to disease. And also, I talked about cardiomyocytes, but that is far from the story. We really need to understand how these cardiac muscle cells respond to signals from endocardial cells, fibroblasts, immune cells. So finding the right scaffolds, finding the right extracellular matrix, and being able to create in vitro platforms that allow us to study the function of the three-dimensional heart is really the holy grail of, I would say, regenerative medicine for cardiac patients. And I'm hoping that the ties that we have in understanding the biology as well as the ability to engineer cells with CRISPR, with reporters, and also working with tremendous engineers to create new biomaterials will actually get us more quickly toward our destination. So with that, I want to thank you for your attention, and my message today at the end is really science equals collaboration. Discovery does not happen without collaboration, and I've had the good fortune of working with many different groups on these projects, so I'll just thank you and end here. Oh, no, go ahead.
Hi, thank you.
Sure.
Thanks, great talk. I had a question about the long non-coding RNAs. Something you'd said about you think they might be linked to disease. Has anyone found either in your lab or in other long non-coding RNA labs point mutations or truncations that are either linked to a disease or linked to susceptibility to a drug or resistance to a drug or any of those things? It seems like a really great area to try and kind of mine next in terms of the genome.
Yes, so the question is about long non-coding RNAs and whether or not they're becoming more recognized targets for disease or disease susceptibility. The problem with studying long non-coding RNAs in this context is they don't code for proteins, so they don't have particular domains that we know where to target or where to look. So unless there is a broad study where every single nucleotide is assessed for function, it's very difficult to match any kind of variant to a disease. We can find variants that do change a nucleotide in a long non-coding RNA in a locus that has disease susceptibility. They're associated with disease susceptibility, but many of these overlap enhancers, right? So one of the things that I didn't mention is the really striking finding that enhancers produce non-coding RNAs. And some of them look a lot like the non-coding RNAs that I talked about. So is that really a function of a variant in a transcription factor binding site or some other binding site that's critical for binding that factor, or is it something associated with the RNA? I would say structural studies like I showed you are going to be critical, because I think, for example, in a 1 kb transcript that doesn't code for a protein, there's probably actually very little of it that's important for function. So being able to identify functional domains will really help us to then kind of retrospectively ask whether or not mutations contribute to other developmental or disease processes.
So kind of piggybacking on the large non-coding RNA section, I know that in bacteria, large non-coding RNAs can bind to metabolites and change their shape and change their function, the whole kind of riboswitch story. So is it possible that that's what's happening here? And could that be a way of kind of integrating, metabolic signaling with more classical signaling pathways?
That would be like my dream come true, okay? So ribozymes are something that obviously have evolved in bacteria, not so much mammals or animals. And that doesn't mean that there isn't some alternate definition of a ribozyme that doesn't exist. And my hope would be that in some way, some of these non-coding RNAs do sense metabolites, which I believe that they probably do. And that might lead to some kind of structural change. Maybe it doesn't have the catalytic activity of a canonical ribozyme, but it may actually elicit other activities that are critical for regulating cellular processes. And so these are some of the things that we hope to discover by looking at a broader set of these structures to determine whether or not they're bound by particular metabolites, because really metabolites are key signaling components to all cellular processes.